Researchers have just reached an important milestone in genetics. They've sequenced the first complete human genome. But wait, hasn't the human genome been sequenced already? Well, yes, but that version, released in 2001, was a first draft, and scientists knew that it contained missing parts and errors. So they've been adding to and correcting that version ever since. Now, for the first time, they have the complete end-to-end sequence for the entire human genome. The new additions make up about 8% of the total genome and amount to a new chromosome's worth of DNA. Some of the gaps in the original sequence that are now filled in include the centromeres, the tightly wound portions of chromosomes that are critical for orchestrating how a cell divides. These regions are also important in human development, certain brain disorders, and neurodegenerative diseases. Knowing the exact sequence of these sections could lead to better treatments for these disorders. The new sequence also maps out the long stretches of repeated DNA that scientists originally thought were junk DNA because they didn't contain genes. But it turns out these regions are just as rich as gene coding parts, and that they do include genes, because they also contain clues about what makes us uniquely human. Understanding these portions of the genome may shed light on what distinguishes us from our closest evolutionary relatives, the primates. Eventually, such a complete sequence will become part of every person's health record, so doctors can mine the genetic information to help people learn which diseases they might be more prone to develop and how to treat them. That won't come for a few years yet, but this is a monumental first step toward that possibility.